Hello friends, how are you doing? And I hope you all are having a great time and you're doing great. Did you revise the last time what we did? And so what did we do? I mean, have you ever opened your books? We were talking about the types of proteins, the different types of proteins. Are they simple? Are they conjugated? Okay. So we are doing the chapter biomolecules. And we started talking about the proteins, and we started our discussion with simple proteins, and we said that yes, these simple proteins have alpha amino acids, right? And then we talked about the conjugated protein, and in this we said these are proteins which will be associated with any other group, with other groups like how we have the the protein associated with phosphoric acid, the nucleoprotein, the protein associated with the nucleic acids, and then the metalloprotein, the flavoproteins can also come, the chromoprotein, all these types of proteins. Yes, the glycolipids, we also talked about the glycolipids, and we said the glycolipids we are telling. The glycoproteins, these are found on the cell membrane and are associated, right, associated with cell antigenicity and the major histocompatibility complex, right. We also classified them according to the function, that whether these are storage protein, as in the case of albumin, right, structural protein, as in structural protein like elastin, then the contractile protein as in actin and myosin, then the transport protein as in the case of aquaporins or carrier protein, then transport of gases protein as in the case of hemoglobin, right, then protein acting as hormones like insulin, protein actings like enzymes, right, all these and many more, if you keep on thinking, the list will never stop probably. Then we started talking about the lipids, and we said that yes, the lipids are microbiomolecules, and we classified them into three types: the simple lipids, the conjugated lipids, and the derived lipids. Okay, we'll start our discussion for today on this note and with this recap, and we'll continue and see how much we can do today as of lipids. So the lipids, we said, can be categorized into, right, the lipids can be categorized into three categories. And what are those three categories? First one we said is a simple lipid. The other one we said was a conjugated lipid, and the third one we said was a derived lipid. Right. So when we are talking about the simple lipid, what will we see? The simple lipids are those lipids, which have a fatty acid and an alcohol. Fatty acid plus alcohol. The conjugated lipids, like they have lipid plus the non-lipid part, and the example we said was lipoprotein, and many more examples, lipoprotein. The glycolipids, anything which you can think of which has lipids as well as the other structure will come in this conjugated. Derived lipids, which substances which are derived from here, and the best example for this we can say, and these will include, not example, these will include the terpenes and the sterols. Okay, terpenes and sterol. I think we did this also last time, but anyways this will refresh your mind. So let's start talking about these simple lipids. Okay, these simple lipids. Simple lipids. The simple lipids can be again categorized or classified into the true fat or the neutral fat or neutral lipids, right? These. So you can write neutral fat or lipids, and the second class, and here this will be the waxes. Now what is a neutral fat? We said that lipids are fatty, simple lipids are fatty acid plus alcohol. Now if this alcohol is glycerol, then that fat or lipid will be a neutral fat or lipid. Okay? If lipids contain fatty acid and glycerol alcohol and glycerol as the alcohol, then it is called as neutral fat or lipid. If the lipids contain fatty acid and glycerol as the alcohol, then it is called as a neutral fat or lipid. This is what a neutral fat or lipid is all about. So now what do we see? Now we will see that how the fatty acid, okay, how the fatty acid, the fatty acids can again be categorized into the essential fatty acid and here will be the non-essential fatty acid. Non-essential fatty acid. Okay. The essential fatty acid and the non-essential fatty acid. In the essential fatty acid, what we will see, these cannot be synthesized by our body and hence have to be incorporated in our diet. These cannot be synthesized. These cannot be synthesized by our body and hence has to be incorporated in the diet. Okay. These cannot be synthesized by our body and hence has to be incorporated in the diet. Non-essential fatty acid, these are synthesized by a body and hence not essential, and hence not essential whether they can be incorporated or not. Whether they can be incorporated, and hence not essential whether these are there in a diet or not. Okay, so these are synthesized by a body and hence not essential for them to be in our diet. Very basic thing, right? Non-essential fatty acid. Now once again, when we are talking about the fatty acid, when we are talking about the essential fatty acid, when we are talking about the essential fatty acid, how many of them are there? So when we are talking about the essential fatty acid, we can linoleic acid, alpha linoleic acid, and the arachidonic acid. Now this arachidonic acid can be synthesized from linoleic acid. Okay, this can be synthesized from here, and hence they say sometimes as semi-essential or things like that. This can be synthesized, okay, this can be synthesized from linoleic acid, right. Now again, the fatty acids can be categorized as saturated and unsaturated. Saturated when they have only single bonds, and unsaturated when they have double or triple bonds. So fatty acids can also be categorized as saturated or unsaturated. Fatty acids can also be categorized as saturated or unsaturated. So this fatty acid these are saturated, have all single bonds. And when we are talking about unsaturated, okay, these have double and triple bonds, right? So saturated having all the single bonds, and the unsaturated have the double and the triple bonds. Now which is more harmful, which is more important, all these we'll get to know as we study them in detail one by one. So now what is a basic structure of glycerol. The bond formation, the bond present. Okay, so if we talk about glycerol, how do we see it as? CH2OH, CHOH and CH2OH, right? And then we are talking about COOHR, right? So this is what? Or this can be written as RCOOH, and there will be the removal. This is what? Fatty acid. So OH. Removal of water molecule, removal of water molecule, and also with this formation of ester bond. Okay, so hence water is, so we can say dehydration synthesis. Why? A water molecule is being removed from here, from this combination of the glycerol and fatty acid. We should not write glycerol here as such, we can write the glycerol here. Right? So CH2OH, CHOH, CH2OH, this is the molecular structure of glycerol, and we combine with fatty acid. Glycerol with fatty acid, this is what? This is the neutral fatty acid, neutral fat or simple fat or simple fat or neutral simple lipid or neutral lipid we are talking about. Why? Because we are taking glycerol into consideration now. So glycerol plus fatty acid, what happens? Here there will be the formation of ester bond, removal of water molecule in us, and also with this formation of ester bond, hence this is dehydration synthesis, as water is being removed. This is dehydration synthesis. Also we can see that maximum 3 ester bonds can form with one glycerol molecule. Why? If we add one more fatty acid over here, right, then again one more water molecule can be removed. Similarly, if we add here one more water molecule can be removed. So with one glycerol molecule a maximum of 3 ester bonds can form. Okay. With one glycerol molecule, a maximum of 3 ester bond formation, a maximum of 3 ester bond can form. Right. Now this tail is a fatty acid. Tail is fatty acid and this is the glycerol. Now this is hydrophobic. And this is hydrophilic. And this is what? Glycerol plus fatty acid. So hence we can say that this is fat molecule. Right? Fat molecule. So since the glycerol or alcohol is hydrophilic, water loving, and tail is fatty acid which is hydrophobic, so we can say hence fat molecules have an amphipathic nature, have amphipathic nature, that is they are both hydrophilic and hydrophobic, that is have both the hydrophilic water loving and the hydrophobic water repelling compounds, right. Now we can also classify the fat into monoglyceride fat, the diglyceride and the triglyceride fat. So fats can also be classified as right, the monoglyceride fat, the diglyceride fat, and the triglyceride fat. Okay. So this is the monoglyceride fat. What will we see here? That one fatty acid. This is the diglyceride fat. This is two fatty acids. And this is a triglyceride fat having three fatty acids. Can you see this over here? Now the saturated fat will be solid at room temperature. Okay, so we can write here that saturated fat are solid at room temperature. Example is ghee. Okay, the saturated fat, while the unsaturated fat are liquid at room temperature. Example is oil. Can you see this thing again here? So the saturated fat are solid at room temperature, example ghee, and the unsaturated fat are liquid at room temperature, example is oil. So friends, you note down this, and after you have noted down, we will continue after a short break. Right friends, so I hope you must have noted down this. So what will we see now is that if we said that all the saturated fat have a single bond and the unsaturated have double or triple bond. So how is it formed? Now glycerol plus saturated fatty acid plus saturated fatty acid will form a saturated fat, okay, and saturated fat are solid at room temperature. We know that, we just said that. However, if we are talking about glycerol or plus an unsaturated fatty acid plus an unsaturated fatty acid, so this will form the unsaturated fat, and this will be liquid at room temperature. Now, by the process of hydrogenation, the unsaturated fat gets transformed into saturated fat. So, hydrogenation is the process by which unsaturated fat gets converted to saturated fat. So, hydrogenation is a process by which unsaturated fat gets converted into saturated fat by addition of hydrogen bonds, which will convert the double and triple bonds, which will convert the double and triple bonds to single bond. Okay, which will convert the double and triple bonds to single bond. However, we will see that the saturated fat is very dangerous. Okay, this is very dangerous, or we can say, rather than using the word dangerous, we can say that not good for our health, as directly deposits, okay, as directly deposits in blood vessels. However, this unsaturated fat, this is little good for our health. Why? Because this unsaturated fat, it will go the conversion, it will undergo the hydrogenation process and will undergo the conversion and gets converted into the saturated fat, So friends, I hope you are following things. We will continue from here the next session. The time we meet again, a very goodbye from this side. Thank you.